Hello A&P students, welcome to the most important unit of your study in A&P, the cardiovascular system. And we will begin our study with blood. And in this video we will be looking at the components of blood and their functions. So let's get started. In medicine, the blood analysis is the most ordered diagnostic test when attempting to determine the state of the health of a patient. Whether a patient is visiting their primary care physician for a routine physical, or a patient enters the emergency room in a crisis situation. The information obtained through blood testing gives us insight into the state of the body systems and whether there is some sort of homeostatic imbalance. A standard blood test looks at the quantity and sometimes the quality of the components of blood and compares them to normal values. This test is so heavily relied on in medicine that it's imperative that any A&P student entering the medical field understand and be able to interpret a blood analysis. And so let's begin that understanding with an exploration of the components of blood. Before we go any further with this video, make sure that you review these concepts from A&P 1. The majority of this material can be found in chapters 2 and 3. Blood is a type of connective tissue, and all connective tissue contains cells and matrix, and within the matrix are fibers that are produced by the cells. When looking at blood, it is hard to see it as a connective tissue because it lacks some of the more obvious connective tissue features. However, as you learn about blood, you will begin to understand its connective function, even though the majority of the time it is on the go. The most common way to study blood is to draw blood from a vein with a technique called venipuncture. That blood is then spun down in a centrifuge for a certain period of time, and the blood separates into three very distinct layers. And they separate out according to their densities, the most dense being on the bottom and the least dense being on top. So let's take a look at the fluid part of blood called the plasma. Plasma has over 100 different dissolved solutes within it, and we can think of it as a solute transporter. And as you can see from this picture, plasma has a straw-colored appearance. And this table from your textbook summarizes the constituents of plasma. So please know what they are and the purpose that they serve. During an emergency situation where there is a significant loss of blood, whole blood may not be available, or a person's blood type may not be known. It is essential to increase the volume of blood so that circulation can be restored. This can be done with normal saline or a multiple electrolyte solution that mimics the electrolyte composition of plasma, such as a Ringer solution. So we took a quick look at the fluid aspect of blood. Now we're going to take a look at the formed elements, or the cells. Now, this table from your textbook does a really good job of summarizing the blood cells and all the information that you need to know about them. We're going to be looking at the blood cells in lab, looking at blood smear slides, so make sure you know how to recognize them. Let's go through the names of the blood cells and then we'll take a look at them on a slide. So we've got the red blood cells, also known as the erythrocytes, the white blood cells categorized as granulocytes and agranulocytes, the granulocytes, which are the neutrophils, the eosinophils, and the basophils. The agranulocytes are the lymphocytes and the monocytes. And then finally we have the platelets, which are not true cells, but fragments of a large cell found in bone marrow called a megakaryocyte. In order to be able to see blood cells well under a microscope, they need to be stained. The most common stain used is Wright's stain, which is actually a combination of two stains. Eosin is a reddish-pinkish dye that is picked up by the proteins in the cytoplasm. Methylene blue is a bluish-purple stain that is picked up by chromatin within the nucleus. Certain blood cells, such as the granulocytes, have granules that are very specific which stain they attract. And this is actually how their specific names came about. The white blood cell whose granules attract the pinkish dye are called eosinophils, whereas the cell that contains granules that are attracted to the blue dye are called basophils. Neutrophils contain granules that attract both dyes, giving neutrophils somewhat of a violet color. And while we're here, why don't we identify the agranulocytes? So we've got a small lymphocyte and we have a monocyte. And the smallest and most numerous blood cell on the slides are going to be the red blood cells. In class, we will talk a little bit about each of these cells, their special characteristics, and their functions. So please read ahead for that lecture. Now that you have gotten an introductory look at the components of blood, let's return to our blood analysis and discuss some of the routine tests that are done to determine the state of health of a patient. The standard tests that are run are the CMP12 and the CBC with differential. CMP stands for Comprehensive Metabolic Panel. Back in my day, the CMP was referred to as the SMAC20 or SMAC25. This is a basic chemistry panel that looks at the function of the liver, kidneys, heart, nerves, bones, and muscles by looking at a select number of plasma constituents and giving some immediate information regarding the physiology of the body. So here are some of the things that we'll see measured in a comprehensive metabolic panel. Such things as electrolytes and minerals, proteins and enzymes, and breakdown products of metabolism. We'll also find measurements for glucose and carbon dioxide and an estimation of the glomerular filtration rate of the kidneys. So let's take a look at our sample blood analysis again and see what we will find. Now don't get too hung up over the details here. You will not be tested on how to analyze a blood test. I just want to expose you to some of the terminology and give you an idea of how revealing a blood test can be. In this column, we see the values for the actual test results. This column is showing the units of measure, and this column is showing the normal values or acceptable ranges. So let's take a look at some of the tests that were performed, and we'll work our way down the line. So we see serum glucose levels are at 82 milligrams per deciliter, and the reference range is between 65 and 99. So this patient falls within the normal levels. And of course, this test is for screening for diabetes or pre-diabetic situations. And the next test that they have listed here is serum uric acid levels. Uric acid is a breakdown product of DNA catabolism, and too much uric acid in the blood can actually cause gout. Now moving on down the line, we see BUN or BUN, which is blood urea nitrogen. Urea is a breakdown product of protein catabolism and is secreted by the kidneys, so BUN levels give us a good idea of how the kidneys are functioning. Creatinine is a breakdown product of muscle metabolism, and so is lactic acid. And lactic acid is also produced by red blood cells because their main mode of energy production is through fermentation. And the next results shown are the estimated glomerular filtration rate, again showing kidney function. Let's move down to these three electrolytes and two minerals that are shown. So sodium, potassium, and chloride are the electrolytes that are being measured here, and calcium and phosphorus are the minerals. Think about bone when you hear about calcium and phosphorus. And you're also looking at endocrine function. Now as we move down, we can see the different proteins that are measured in the plasma, such as albumin and globulin. Now, albumin is produced by the liver, and it's a very important component of plasma in that it is the main contributor to osmotic pressure. Now the globulins are a class of proteins that have a number of different functions, and some of them are produced by the liver, and some, the gamma globulins, are the antibodies that are released by plasma cells. Let's move down now to the enzyme group, starting with bilirubin. Now, bilirubin is not really an enzyme, even though it is grouped here with them. It is a breakdown product of heme catabolism. The heme is separated out from hemoglobin when red blood cells die and get processed in the spleen. Bilirubin is used by the liver as a component of bile, and increased levels of it in the plasma can be caused by liver or gallbladder disease or anemia. Moving down to the last three tests on this page, we have three enzymes, starting with alkaline phosphatase. Alk phosphatase is an enzyme that's used by the liver and bone, and increased levels in the blood could indicate bone disorder or could actually indicate pregnancy. Lactate dehydrogenase is an enzyme used by all cells and is released during cell trauma. So an increased level in the blood would indicate increased cell death. AST is an enzyme that's used by both the heart and the liver, and we'll see big increases of this enzyme during heart attacks. There are a few more enzymes on the second page of this report, but I think you get the drift here. The information gained by just this basic chemistry panel is huge, which is why it is a routinely ordered group of tests. The second part of the report is the CBC with differential. CBC stands for complete blood count, and differential means a differential count of all the white blood cells. Let's look at page 3 of the report to see the results of those tests. So I'm just going to run through these fairly quickly. You can read along with me. We have a total white blood cell count of 5,300, a red blood cell count of 4.28 million, a hemoglobin value of 12.4 grams per deciliter, and we have a hematocrit at 39%. Then we're going to move down to platelets, and we see we've got 265,000 platelets. We have neutrophils at 53%, lymphocytes at 34%, monocytes at 10%, eosinophils at 2%, and basophils at 1%. And let's move down now to the absolute numbers for the white blood cells. So neutrophils came in at 2,800, lymphocytes at 1,800, monocytes at 500, eosinophils at 100, and basophils at 0. Immature granulocytes are at 0%, and the absolute number for immature granulocytes is 0. Nucleated red blood cells, none seen. And just in case you didn't notice in the units, these absolute numbers for blood cells is per microliter. So that'll do it for this video. In our next blood video, we will look at these components a little more closely and talk about some of the physiology and some of the disorders. So until then.